My name is Christopher Clemsen, and I'm research group leader here at Center for Basic Metabolic Research. It is my real honor and pleasure to introduce the final speaker of today, Professor Richard DiMarchi from Indiana University. So Richard is a distinguished professor and chair in biomolecular science. And Richard has demonstrated throughout his career that you can effortlessly transition between industry and academia, having held positions both at Eli Lilly and Novo Nordisk, the two powerhouses in metabolism research, as vice president, and has held professorship with Indiana University for decades, also published several hundred papers, and also co-inventor of more than 100 patents, has been a founder of more than, or perhaps around 6, and keep in mind, successful biotech companies, so meaning biotech companies that has actually been acquired or making big partnerships with pharma. So really long, have received numerous awards. And I think in my book, I'm most impressed by your work on inventing basically the co-agonist molecules that we're now seeing entering the clinic for treatment of metabolic complications and probably beyond. And Richard is a really outstanding scientist and generous collaborator, so it's an honor to have you here, and I look forward to see what you have to share with us today.
Thank you, Christopher. It's been a spectacular day of cutting-edge biology, and here as a chemist, I feel like the guy who arrives at a black tie event in a brown suit. Right. And my job is to try and translate how we use chemistry to interrogate biology, not just to make drugs. There's a difference between synthetic chemistry, medicinal chemistry, and what we now call chemical biology, and hopefully I'll give you examples of each of those. You can see these molecules that I have lived with over the course of 40 years, and to some of them, you'll know them as drugs. They take on this magical quality. But I can assure you that when we first made these molecules, they were just chemicals. They were just one of many peptides that were in the lab that somehow evolved and graduated to be these magical substances. Some are still on their way. I put in the middle of the slide this textbook from K.C. Nicolau. Many of you would not know him. He comes from the rich tradition of Bob Woodward, E.J. Corey, as true, hardcore, synthetic chemists. And in that book, there's over 30 chapters on miraculous molecules. All of the peptides, proteins, antibodies, antisense are captured in one chapter. It is the way that the world has looked at what constitutes a drug. Large molecules were boutique, right? Because we were trapped by the fact that we couldn't make them because of their complexity. What chemistry gives us is the control over physical matter, which gives us control over the biological matters. And when you look at these drugs with traditional chemistry, you can see it's very different from what we know from the 20 basic amino acids that are the building blocks that we use to create higher order structure, right? Secondary structure, tertiary structure is what gives peptides, proteins, their incredible potency and selectivity, whereas this does this with much more synthetic diversity. And the two have largely lived apart from one another because these were designed to be oral drugs with molecular weights less than 500, where we have worked in a space that is larger and typically injectable substances. Now, I left New York in a very prominent lab that I'll tell you about in a second to go to Lilly in Indianapolis. The doorman in Manhattan asked me, "Why are you going to Indiana?" He says, "There's only two reasons. You have trouble with the law, or you're crazy." And I went to learn the technology. I didn't go to produce human insulin. I went because I saw what this represented. And you would know that the landmark publication was in 1978 from the City of Hope in collaboration with Genentech, Arthur Riggs, and Dave Goodell, which was only a few years from those landmark publications in PNAS that came from Paul Berg and Conan Boyer talking about the ability to splice genes. I tell students that I go back to the BC period, right, before cloning. We were extracting these proteins from urine, from blood, right, and from glands. And in fact, one of the more colorful ones was called holy water, right? This was follicle-stimulating hormone, of which the Catholic Church got very much involved in. It would take 10 nuns, 10 days, to produce enough follicle-stimulating hormone for one treatment to one woman. And the production of insulin came to us from Genentech, where they had produced not much more than a milligram, and the launch quantity in 5 years was 100 kilograms. Now, if you've come from Malmo and call that a milligram, that distance, going from a milligram to 100 kilograms is roughly a journey to the moon and back 10 times. It was pure insanity. When I went to get the cyanogen bromide that was needed to process those fusion proteins, I was told the world has never made that much cyanogen bromide. And if it sounds as if it's toxic, it is. And even worse so, I had to make it from cyanide and bromine to be used at 1,000 liters every other day to make this protein. It was insanity, and most people focused on the fact that we now had the human sequence. We had changed this alanine to a threonine. It's a methyl group to a hydroxyethyl, which is not much of a change. But it had been believed that that change could be part, that is, that alanine change in porcine insulin could be part of the reason why diabetics suffered the consequences of the disease. And the more important part, if you go back and read Irv Johnson's book, who had nothing to do with metabolism, a real visionary fellow. He was a research oncologist that saw the power of biotechnology. It was about the fact that the growth in those needing insulin was 5% per year, and the growth in the glands to produce that insulin was -1. We were moving to a point where we would be rationing insulin. And God love us. You know, we got this technology at just that time that we could produce the amount of material that was produced. The reason why it got approved so quickly is the fear that there would be an environmental contaminant, right? So that somewhere in the glandular process, we'd be treating our patients with an adventitious biological agent, and that was a huge fear for the agency. If you remember, growth hormone deficient children were appearing with CJD disease, right? Prions from the growth hormone extracted from the brains of cadavers. I went there as a chemist. I saw in this the power to be able to shuffle nature's amino acids to produce something that didn't exist. And if you think about the history of medicinal chemistry, we get penicillin, we make better forms of beta-lactams, right? We get vinca alkaloids, we modify them to make better drugs. And I looked at these proteins as natural products, and you needed the power of this methodology to scale it. And Humalog, Lispro insulin, was the first that was optimized for this purpose. But with incredible delicacy, because we had waited 60 years to get nature's sequence, human insulin. What makes you think that you, as a chemist at roughly 30 years of age, could design something better than nature itself? So this comes back in the whole discussion, because right on the heels of that comes this publication from Graham Bell, who had been at Chiron in cloning of the human pre-pro glucagon gene. He was on his way to the University of Chicago. He preceded me in leaving a biotech or a pharmaceutical company to go to academics. And he describes this sequence where you have glucagon, GLP-1, right? Glucagon-like based upon its homology, and glucagon-like 2 in the human sequence. And he suspects that the GLP-1 sequence is this 37 residue peptide, because you have this processing site. Let me see how I point with this thing, right? This lysine arginine here, common processing site that should give you a new N-terminus, because that had all been worked out with these lipotropins about these doubly cationic amino acids. But for those that had been familiar with the insulin, with the glucagon sequence, it was curious that if that's the N-terminus, glucagon starts here, and even more so, 4, 5, 6, 22, 25 are homologous to glucagon. So why would you be processing here? Why did nature choose to extend the molecule by 6 amino acids to get an active substance? And if I can move this along, you'll see a picture of Bruce Merrifield. I don't know if you can move it along up there, but just go ahead and do it. And Bruce Merrifield was that laboratory in New York where I had been working. He was the father of being able to synthesize peptides on solid support. And his idea, his methodology has subtly changed, but the concept is still the same in how we produce peptides for research purposes and for commercial purposes. And I'm very, very humble man who was sort of beat up by the inventiveness of what he did in changing synthetic chemistry to a point where you did not characterize the intermediates. Where's your carbon-nitrogen analysis? Do you have crystal structure on all of your intermediates? He was a biochemist by training. And the molecule that was most controversial was glucagon. So Svetlana Moishev was a graduate student in New York at the Rockefeller when I arrived as a postdoc who had worked on glucagon because it was the tip of the spear in synthetic chemists arguing that solid phase could not deliver high-quality peptides. And I'll show you in a minute why that is. She then left with her husband, who was doing a fellowship, his internship and fellowship as a physician at the MGH, to take a job in a core facility to make these peptides. And she was uniquely positioned to work on these substances that looked like glucagon. And aside, if Jens is here, Jens was doing the work here in Copenhagen. She was doing the work over in Boston. And it's still controversial as who did what, when, and I'm not going to go in there. That's not my lecture. Jens, you've done phenomenal work, right? You've been a high priest. I've respected and learned from what you have done. I'm just delighted that Svetlana is now part of the discussion. So as you go forward, so here were these two incretins, GIP, that had been discovered in 1971, right, by the Canadians, not in Toronto, but on the West Coast. And it had proven to be, well, let's call it a failure, right? Its translation into treating human diabetes did not work, despite being a physiological incretin. And of course, in 96, we find that GLP is an incretin. Now, for all of the beauty and the virtues of GLP-1, Jens did some really important work with Michael Nock and Werner Kurzfeld, the 1993 publication in Diabetologia, the 4-hour infusion with native GLP-1 showing its efficacy in type 2 diabetics was of instrumental importance to us. But it's not a drug. Let's be very, very, very clear. It's too unstable. It's too short-acting to be a drug. It required chemical intervention. And we went off on this side journey that was driven by the Amylin Corporation when registering the first GLP-1 mimetic, which was this peptide derived from Gila monster, right? Exendin 4 Byetta was the substance that had higher stability and somewhat longer duration of action so that it could be used as a twice-a-day medication. And in this work that goes back to 1992, you can see the publication from John Ings, 92. This was 2002. The medicine was registered in 2005, working in all forms of type 2 diabetes when used as a prandial medication, twice a day, 10 micrograms, I think, at each of those doses. At Lilly, we focused on making a longer-acting substance and steered away from the Gila monster derived because we had been schooled, don't make changes in the sequence. You're going to run into problems. If some of you remember, there were a couple analogs, insulin analogs, that were made here, very elegant work from Jan Markussen, that ended up causing problems in their development. Carcinomas in rodents, skin reactions in patients, which made everyone fearful that you better stay very close. And you can see up above, the ascendant sequence in red is quite different from human GLP, right? It's less than 50% homologous with the C-terminal tail that we call the CEX, the C-terminal extended sequence, which is a tryptophan cage that folds back over the molecule because there's a tryptophan at position 25 that is buried under that tail that folds back over it. And that hides the hydrophobic face that gives the molecules more physical stability and more solubility. And what you see here was the emergence of what became Trulicity. This was the FC fusion that we made at Lilly that started on my clock, and they continued after I left. And it was, I think, registered for treatment of type 2 diabetes in 2014, 3 years before Ozempic. So this was the first engineered molecule to have once a week time action, and it was it is a very good drug for treating hyperglycemia. Okay, now, where does obesity come? Because this was sort of a derivative of the work that had been done on diabetes. And in 1996, Sir Steve Bloom published this paper on the ICV infusion into rats for their ability to suppress food. That was the state of the art. That's how you get into nature in those days. Suad Efendik and I had struck a collaboration when we were working on Lispro insulin development. That's how I got exposure to Scandinavia, and I was so impressed by the willingness of the community to do clinical research of the type that has characterized Jens' work, Steve Bloom's work, what Suad Efendik was doing, what Creutzfeldt and Nauk was doing, because I am a great believer in vitro, in vivo, in veritas, in truth. Until you put it in a patient, I don't believe it. And so I had encouraged Suad, who had kept telling me this food suppression that we were seeing in rodents, anyone who was working with GLP would see it, proved to me that it was going to work in overweight diabetics, because remember, GIP had failed when we put it into diabetic patients. And indeed, this became part of a patent application that was filed at the end of 1996, that year. And it issued with claims 7 years later that covered the whole class of GLP agonists for treating obesity. This is for obesity, it's not diabetes, right? The amazing thing when I was at Lilly is they were not interested. Obesity is not a disease, and even if it were, people are not going to take injections to treat that disease, okay? It was that simple, and furthermore, the whole fen-phen scare were concerning large corporations. There was too much litigation and liability surrounding treating this disease. And if that wasn't enough, it was the failure of leptin to translate that said, maybe you'll get a couple of pounds of weight loss, but you're not going to have the transformative effects that we came to see in Ozempic. I'll show that in a second. So there is the wonderful work that has been done here at Novo, influenced by University of Copenhagen and Lotte Knudsen, Thomas Kruse, Jesper Lau, Peter Kurtzhal, with the tie back to Jens Markussen, and the whole start of this fatty acylation that I've often called lipidation to bind plasma proteins. And you can see that the improvement is meaningful, clinically significant, and statistically significant. Note that the decrease at 4 weeks is almost spot on from what Suad Efendik had seen in his infusion study. And for the importance of this work and the work that Novo did in showing that you could register a drug to treat obesity, this, to me, was still a diabetes drug that lowered your body weight. It isn't the transformative drug that we were waiting for, which came years later when, again, Thomas and Jesper, it's always hard to know who did exactly what, changed the linker in the molecule, putting in a small polyethylene glycol linker. They added an acetate, that is, they turned the C16 fatty acid into a diacid to extend its duration of action. That was the primary purpose of doing this work, to get to a once-a-week drug, and it's a very good once-a-week drug. But the real aha the sort of thing that I say in veritas, right? Until you put it into humans, that's the spectacular. And I would say serendipitous finding, because based on rodent pharmacology, you would not predict that this molecule would be 10 times more potent than Lyra and at least twice as efficacious in lowering body weight. That was the one that got everybody's attention, because we were all wondering whether we could get beyond 10%, not to 15%, which this is showing, right? You know, half of the subjects are showing as much as 15% weight loss. That was really a benchmark discovery for this whole metabolism community. And right on the heels of that, here comes Lilly to spoil the party, right? This was a tough day in Denmark when, at the EASD meeting, that Lilly reports Tirzepatide is now causing as much as 20% body weight loss in half the subjects, and not just obese diabetics. These are non-diabetics. These are the diabetics who are still showing appreciable weight loss, to the point where it was debated what is GIP doing. It's still debated, right, with the Amgen 133 and antagonist, which is quite a rogue, quite frankly. And so my journey in all of this was in academics. I had already left Lilly. They weren't interested in diabetes, and I'll show how we came back to this in working with Matthias Chupp, who had just come from Potsdam to Cincinnati and was willing to do experiments with me to see if we could find some new mechanism for doing this. And you'll know that last year at the ADA meeting, Lilly published on the triple agonist that we had pioneered, where these subjects are losing a pound per week for a year, and more importantly, using glucagon as it was never meant to be used. And so the nexus, the start of the story, was when I went to Indiana, leaving Lilly. I said, we've got to make a better glucagon. I mean, this is a life-saving drug every bit as much as insulin is, even more so because you use it at a time of acute need. You're unconscious or semi-conscious from an overdose of insulin, and what you get is a chemistry kit. You get a little powder, you get a diluent, you get a syringe, and you're supposed to put all of that together and give yourself an injection. Lilly was not interested in investing in making what I called the equivalent of an epinephrine pen that you'd have to immediately use. Novo was not interested. Even the JDRF, Juvenile Diabetes Research Foundation, turned us down. And so we took it on nonetheless with a postdoc and a graduate student in the laboratory, and this is the collection of the chemistries we advanced for dealing with problem peptides that look like glucagon. And the historical problem resided right, which position, 15, 16, right there, right? That ASP-SEER sequence is labile in acid rearranges to what's called an aspartimide in the peptide cleaves. And so when you were making these synthetic peptides in the day when Svetlana Moishev was getting started, that was an insurmountable problem, in addition to some deamidations and the fact that glucagon is just not soluble in aqueous solvents. You saw that the diluent that you use is 0.01 normal hydrochloric acid. Right? It's very acidic solvent. Compound's not stable. You're told to solubilize it, inject, throw it away. And we went about demonstrating that when you put an AIB not at position 2, as you've seen it for DPP4 protection, but in the middle of the molecule, you could stabilize this sequence, and then you could use changes in isoelectric character, decreasing the PI of the molecule to get molecules that were soluble, stable, and this company, Marcadia, was off and running with this and gets distracted by the whole subject of obesity, as I'll show you in a minute. But we also introduced the concept of hydrophilic aromaticity, using isoacyl bonds to create a reversible conversion from a soluble to from an insoluble to a soluble substance, and also using isosteric inversion to give you molecules that allow you to do the sort of pharmacology that Matthias demonstrated in one of the first experiments where I asked him, now having something that was physiologically stable and soluble, just infuse it for 2 weeks and see how it does relative to Ascendant 4. And we were shocked when on a mole-for-mole basis, it was every bit as effective, and we did not see an increase in glycemia, right? The hallmark of what glucagon is supposed to do. And you would know that this was the beginning of, could we put these molecules together and defy what the logic was. Here's your publication, too, from 1994, which was an important publication. In that point in time, nearly 24 molecules were made, half in GLP, half on the glucagon sequence. And what they showed is that nature selects for specificity. And when you start to change the sequence, what you most often see is a loss of potency. You don't see what we had here, where John Day, now at Lilly in their insulin program, took a molecule that was as potent at the glucagon receptor as it was at the GLP receptor. It was a co-agonist, sort of a master key that would open both doors, both receptors, equally well. And when Matthias tested that molecule, it was published in Nature Chemical Biology in 2009, it was more effective. It's the blue column and lowering, the blue line and lowering body weight, and it did it without further suppressing food intake. It was primarily by increasing energy expenditure. And so we said, this is attractive, right? We could use GLP to buffer against what glucagon is capable of doing and have two different mechanisms for lowering body weight. But for 20 years, led most notably by Roger Unger, the world had been focused on glucagon antagonism. And here we're talking about agonism. And the way this landed with the advisors was, you're just one of these American tourists in downtown London driving on the wrong side of the road. You're going to crash. And yet you know now that BI and Zealand are working with a co-agonist where they've shown lovely results on liver disease. And Phil Shearer just last week published about the importance of glucagon at the kidney. So it just shows you, here we had this hormone, glucagon, for 80 years and characterized it as a counter-regulatory hormone. Let's be very clear. There's nothing counter about this hormone. It's a regulatory hormone every bit as much as insulin's a regulatory hormone. It's just that the success and the glamour and the miraculous properties of insulin had minimized glucagon, and we didn't have a form to really study its biology. So back to GIP and this observation, again, coming back to Jens and his colleagues' report, that the reason why GIP doesn't work is because these folks are diabetic. High glucose, if corrected, could restore a response, but GLP had maintained a response in the face of hyperglycemia that gets better as their glucose is being improved. Could you use the GLP as the first rocket to ignite the sensitization to allow GIP to do its thing so that you get the best of both worlds in recapitulating physiological incretin control? Now, let me be very clear. When we started this work, we were not focused on obesity. We were thinking glycemia because both of these were glycemic agents. And when we put them into DIO mice, what we saw was GIP did virtually nothing, and the combination did no better than GLP alone. Obviously, which one would you pick to treat disease? Until you got out almost to a week, and all of a sudden these lines started to segregate. And this was reproduced several times, and it just got better and better the longer you treated these animals. So jumping ahead, you would know this was the beginning of what became Tirzepatide. In many ways, people have said to me, they borrowed your concept of co-agonism, and they borrowed Novo's diacid to generate a molecule that gives outcomes that are superior to Ozempic. This study was published in 2017. It was to be a Roche study. They decided they didn't want to be in metabolism after having paid $250 million to buy the company. I bought back the assets on a loan from them for $5 million, personal loan, right? If you're going to do that, you really have to believe in your hypothesis, and you have to have a very understanding spouse. And indeed, in this experiment, we had an open label reference because I wanted to make sure we didn't have a funny study effect with a dose titration that had been optimized for this drug. And we just had one dose of our compound. We didn't have the money to do much more than that. And what you see is the compound was very effective relative to what we knew for Victoza at that time, but did not segregate statistically significantly given the power of the study. But going back to what we had seen from the Copenhagen work on the importance of being in a glycemic control, if you do a post-hoc stratification and look at those patients that were in better control at the start, you can see that what was then an NN compound, because Novo bought the assets and I was associated with Novo in finishing the study. I have to tell you, that was the last place I ever thought I would end up. And when I welcomed them into Indianapolis, I became less than welcome myself, but it's been a spectacular association, some really great people. And you can see in that post-hoc stratification, the glycemic control was much better, and the body weight was much better. But it was not long thereafter, as I said, that Lilly publishes these results with Tirzepatide. And basically what's happening here is you're going from what I just showed you to Tirzepatide with the diacid. It's doing for the co-agonist what the diacid did in converting Victoza into Ozempic. Really incredible results with, again, more than half of the subjects losing more than 20% body weight. If you want to see what that looks like, you can Google it. I'm sure some of you do. When I was giving grand rounds at Northwestern, I think it was in November, this one gal comes up and gives me a big hug. She said she's part of the 100 Club, people who have lost more than 100 pounds. 53 years of age, still 190 pounds. I can't show you her face. This is somebody else who's on the internet. And she said to me she was 310 pounds and had not been less than 200 since she was in her teen years, okay? Her quote was, "I got a get out of jail card." You know, the Monopoly game. It was that phenomenal. And so where do you go from here? We made the triple agonists, and if you know how to Rubik's Cubes like, it works that way for a chemist too. You get one face right, you screw up the second face. You get two faces right, you can't get the third face in. And it was, as the chemists would know, the use of the fatty acid as part of the pharmacophore, right? Because a fatty acid is not just going to wave out in solvent. It's grease. It's got to sit down somewhere. It sits down on the most greasy part of the peptide. So where you put that fatty acid affects your ability to have either selectivity or to generate polyagonism. And indeed, you have to be really careful because you can engineer in promiscuity that you did not want or did not know was there. But we made these molecules. We showed again with Matthias Brian Finnan, who was a student with me and then a fellow with Matthias, that it had this increased ability to delipidate the liver. One of the common observations in all the work that was done with glucagon antagonists is that we were lowering plasma glucose and increasing hepatic fatty stores, right? Not a good trade-off. And so this was delipidating the liver, and you know what Lilly published in June, right? So here we are, right? The market cap of Lilly and Novo is equal to the market cap of Merck, J&J, Pfizer, Novartis, Roche, and Glaxo combined. And it's 4 peptides, right? We used to hold sessions at the peptide symposium that said, can peptides be drugs, right? This is liraglutide, semaglutide, dulaglutide, and Tirzepatide. that have generated all of this incredible market cap. And if you remember back 30 years ago, if you were there was the Josh Bozier story in leaving Merck to start Vertex, the billion dollar drug, right? So advance 30 years. Now it's the $10 billion drug that everybody is focused on, including Bernie Sanders. Okay, so take note that people are watching how profitable we become, but we as scientists continue the quest for the perfect drug. And that's where I'm going to close by telling you just a few stories of where we're going. And if you'll have me back in the fall, I'll tell you even deeper about what I think is the next generation of science. But a taste of this was the fact that at Lilly, I had developed a Forteo, the N-terminal fragment, 34 residues from PTH, which had been with us for 70 years, right? I mean, this was discovered by Collip in the days when insulin was being discovered. And we demonstrated that if you gave it as a subcutaneous injection every day at 20 micrograms, you could build bone. Here's a woman roughly 2 years after starting therapy, and her bone structure was no different after therapy from her daughter of 30 years younger, 90% reduction in fractures in those clinical studies, a truly regenerative drug, a bone-building drug, but dependent on pulsatile injection, very much like a prandial insulin. And where we were focused on was hypo-PT. I had always wanted to take this drug and put it in a pump for this rare disease. Lilly was uninterested. You get the message as to why I left, right? We're uninterested because it was too small an opportunity. A physician, Gordon Cutler, who had been at the NIH, went on, took the drug, and demonstrated that if you just infused it, you could control bone metabolism, right? Phosphate, calcium, to put these subjects in a healthy state. And so what we did is we developed dipeptide-based prodrugs, something I had introduced Novo to, which was if you know the structure of a peptide, right? Dipeptides exist in the trans configuration. How do I do that, right? And so when you move them to the cis configuration, you're moving that N-terminal amine, the nucleophile, right next to the carbonyl 2 amino acid south, such that it can cyclize to a 6-membered ring, a diketopiperazine. That's known in peptide synthesis and organic solvents, but what we showed is that while your peptides and proteins are stable under physiological conditions, if you stabilize the cis configuration by simply introducing methyl groups into an alanine or a methyl group into a glycine, making it sarcosine, you can convert that speed, which doesn't happen at all in your native peptides, into something that can happen with a half-life of 10 minutes. Now, for the chemists, think about that. This is aminolysis. Nature uses enzymes. DPP4 cleaves dipeptides, and here we're doing it with chemistry in an intramolecular reaction, which means that it's concentration independent, all under physiological conditions, so that I can then build a prodrug on PTH that when given once a week to patients, these happen to be volunteers, but the compound's in phase 2 clinical trials, you can see you can get this very slow onset and this incredibly flat response where the therapeutic index is much improved. Because what they need is replacement of basal PTH. They've lost their parathyroid gland, usually because of surgery. It's sort of the basal bolus concept. And in these subjects, they are being anabolically treated by this infusion, as Gordon Cutler had shown in his pump infusion. Okay, so obviously, what can you do with an incretin? So this is in monkeys now, but what we did is we refined the chemistry, adding a new twist to it that I can't share with you because it's proprietary. But here is Tirzepatide in a cynomolgus monkey, which clears these lipidated compounds 3 times faster than a human does. Half-life of 50 hours in a monkey, 150 hours in a human. Tirzepatide, exact same phenomena. It's due to the accelerated clearance of albumin in a monkey relative to a human. You can see that after a week, its concentration is about 10% of where it was, and if you extrapolate out, it'd be at 1% after 2 weeks. And you can see the very slow onset by using these prodrugs to release drug. This is the drug form that, again, gives you a very flat response that's going out into 3 weeks for a monkey. And we would hope that that would give you the ability to minimize the GI effects or to dose more intensively since you don't have the fluctuations. Many of you wouldn't know that after a single sub-Q injection, the coefficient of variability is as high as 40%, right? And that's why the dovetailing is incredibly important, because it minimizes that coefficient of variation, and it became an important part of the ability to develop Ozempic semaglutide into Rybelsus. And so as I move to close, everybody's got their favorite addition, whether it's GIP, glucagon, GIP antagonists, NK2 agonists. You know, pick your poison as to how you're going to take this 20% response to something that's closer to 30 or 40. You know, for all the excess and the celebration we should have, remember a common definition of obesity is 20% excess body weight. So we're at the end of the beginning. You know, we're just minimally meeting the needs of those who have been diagnosed with obesity, and what can we add? And remember that it doesn't have to just be a peptide, right? We've done a lot of work with proteins, and some of them were on that first slide that I shared with you. And I'll just remind you of something that we published previously, now going back to the first company that I had started, where we had shown at Lilly that if you mutated the tryptophan at position 100 in leptin with glutamic acid, you made a monomeric soluble substance that you could crystallize, and it was published in Nature way back when. And then what we did is, on top of that, we used amber suppression to incorporate a non-native amino acid, right? The Ambrex was called Ambrex because it was suppression of the amber codon to put in functionality that you only saw in those early pages, right? Now, can we put ketones, aldehydes, olefins into proteins in a way that nature never did, so we can create more function? And J&J bought Ambrex in January for, I don't know, $2 billion, because it gave them the ability to site-specifically label antibodies with cytotoxic agents. In this instance, we were searching for a location where we could put a protractor. And the importance of it is in a paper that, where's Christopher? It was your publication, my friend, when you were with Matthias. And what it showed is that if you took DIO mice and you treated them with leptin, they did not lose weight. In fact, they slightly gained weight relative to vehicle, that has led to some discussion over, do we want leptin antagonists instead of agonists? Here we go again. But if you used one of the co-agonists, in this case it was a GLP-GIP co-agonist, when the animals lost roughly 15 to 20% body weight, all of a sudden the leptin started to work, right? Very reminiscent, what I showed you a minute ago, of GLP releasing the brake on GIP to give you greater pharmacology. And we thought that this was because the animals were at a lower weight. They were now leptin sensitive. But when the subsequent experiment was done, I think that was Timo Mueller's publication, and we diet restricted, paired them back to this point, they did not respond to leptin. It was a pharmacological sensitization. And the investors at Ambrex looked at me and said, this is great stuff, publish it. But how do you propose to decrease body weight in humans by 15 to 20%? Because now we're back in 2014, right? So there is this open question whether humans will respond. Is 15 to 20% the right level? How would you construct the leptin, so on and so forth? So I'm going to end here, and we'll answer this question if you'll have me back in the fall. This is the sort of question you've seen in study sections. It's the sort of question you see in every research management group, and you see it with venture capitalists as well. What we need is a brand new idea, something novel, never thought of before, that has been thoroughly tested, right? How many times have you been told, great idea, but do you have data, right? In God we trust, all others bring data. So that's what we need to discuss, is to how do we get from this spectacular accomplishment that everyone should celebrate, right? We're now treating excess weight the way we have treated glucose, lipids, hypertension, and it wasn't supposed to happen, okay? I mean, just look at a century of history with thyroid hormones, amphetamines, DNP, right, dinitrophenol. Let's just hope everything holds together, right? Because the stuff is being distributed in large numbers, and you know it's being used in ways that it was not tested in regulatory settings. So let's just hope all continues to go. Thank you very much for the invitation.
Thank you, Richard, for fantastic and thought-provoking overview, and thank you for ending on high note with showing one of my postdocs. Now the floor is open we'll do some questions before Julene will end off, but I'm sure there's a lot of questions.
We have some time. Did I do okay? Oh, great, good.
There's one up here, and there's one in the back. We start in the back, maybe. There's a microphone already.
Hi, it was fantastic talk. I have a question regarding those peptides, because there is receptors for those peptides on immune cells. Is industry actually investigating the role of those peptides as they become more complex by binding leptin with GLP-1 and so on the immune response, for example, in the settings of cancer, infection, autoimmunity, and so on? So maybe you can comment on that.
No, it's a great question, and you're asked that question every time someone looks to make an investment, whether it's venture capital, whether it's partnering with a pharma company. How do you know that it isn't going to prove to be immunogenic, right? I mean, if you remember the history before biotechnology, most of these substances that came from other species were highly immunogenic. In fact, pegylation came to us as a means to suppress immunogenicity on natural sourced proteins, and then from there we began to see that it changed the pharmacokinetics. That was a serendipitous observation. What you do is use the predictive algorithms that you have. You expose human lymphocytes in culture, and you try and maintain the physical properties of the molecule to be as monomeric as possible, because aggregation and certainly precipitation at the site of injection is a great way to call in the immune response. And then you pray, you know, because humans are outbred, and you just hope that you don't run into a stop signal. Again, the Amylin Corporation ran with this Gila monster sequence that was as foreign as can be, and I think the folks at Novo, probably Lotte, led the charge that says we're not going to deal with something that foreign. We're going to make something that is gently modified, which became liraglutide. And at Lilly, we were doing the same thing. And to Amylin's credit, whether they were smarter than us or they just luckier than us, they didn't run into neutralizing responses. But when they decided to make a depot formulation of this, I said, wow, that's really nutty. And it didn't fail. For immunogenicity reasons, it commercially failed because they used a 26-gauge needle, which is damn painful if you have to use it. And it also was not anywhere as good as Trulicity and then later Ozempic. So the point of the matter is there are some high-tech ways to try and predict and to minimize it, but there's still a certain amount of empiricism that you have to do the experiment and hope for the best.
We have a question up here also.
Yep.
Thank you for the fascinating talk and your fascinating work. What are your thoughts about using the GLP agonists for treatment of type 1 diabetes? I know there's some hint that Ozempic is actually good for type 1 diabetes in early stage.
Well, I was captured by another publication from Jens, where he had shown that the GIP was stimulating glucagon when blood glucose was low. I mean, that's what we would love to have at this point in time, is a hormone that does for hypo what GLP-GIP does for hyper, right? That was limiting, because it'd be the ideal substance to mix. If Jens, if I'm right, it didn't seem to work in diabetics, right? In those, you just didn't get the glucagon kick. So that's unfortunate. Now, can you still get the anabolic effect at the pancreas, the sparing of insulin? Yeah, as long as the subject has functional beta mass, they're C-peptide positive, you'll get some bump on this. And if that can move somebody from 30-40% reservoir to 50-60 or better, more power to you. I would think it's advisable. Now, let's be clear, I'm not licensed to practice medicine. So you might want to speak with some of the clinicians like Jens.
There's a question up here from
So very
colleague.
nice talk.
Mads.
Yeah, good to see you again. So I have a question. So I know that you're a chemist, so this is probably a trick question. It's not, because I really want to know, so I don't know the answer to this. So you talk about bringing back the effect of GIP by introducing GLP-1. But could you speak to sort of the different efficacy of both Tirzepatide and semaglutide in people with and without diabetes that are obese? So why are we seeing this maybe almost a 50% reduction in efficacy?
Well,
type 2 diabetes.
So let me give you my hypothesis, and I'll even comment on the Amgen work if you wish, in looking at antagonists. So what I see is the difference in the case of Mounjaro versus Ozempic is there's some additional weight loss, right? And I'll let Lilly and Novo debate it as to just whether it's 1%, 2%, 5%, whatever it may be. In fact, we probably don't have enough results toe-to-toe, side-by-side, for the long enough period of time to say with certainty. But it appears as if there is an ability to sustain more efficacy in diabetics that are obese with the co-agonists than there is with the monoagonist. That's where the difference seems to increase, and the glycemic efficacy of GIP tends to show itself. Okay, Novo's racing to add amylin. Thomas, I often say to my students, if you think you're really great, come talk to me after you've discovered your second drug. Anybody can win the lottery, all right? But when you've done it twice, then it's not an accident, because lightning doesn't hit you twice. Thomas may be one of these fellows from his work on semaglutide and now yeah, right. Amicretin, right? You call it amicretin, right? So what I'm saying is this. There are different ways to get there. If I may, the only thing I actually embrace the controversy around the Amgen AMG 133. I think, is that the right number? Because I was so attacked about glucagon agonism when we were supposed to be doing antagonism, right? Let's embrace the fact that we don't quite understand what's going on here. It's provocative. Let's get more data. I am disappointed that they've not made the reagent available to the scientific community. If you're going to put out provocative data like that, then let us have the reagent to determine, is it real? What's causing this effect? And if you look at table, I think it's 1C or so, you'll notice that their IC50 for antagonism is nanomolar, and their EC50 for agonism at GLP is picomolar, every bit as potent as dulaglutide, Trulicity, is as a GLP agonist. I don't understand why we're dose limited to 8 milligrams per month with this substance, and something that's equally potent at GLP, structurally similar, can be given at 800 milligrams. You would think that would cause a phenomenal adverse gut effect. Data talks, right? Those are the results. How we interpret them is our part, right? But the results are there. I want to see it reproduced. If it does work, I've got a working hypothesis as to how this could possibly be true, that agonism and antagonism may do the same thing, and it's not this sort of LHRH super agonist giving you an antagonist. But we can discuss at the break. Good to see you.
We'll take the final question up here.
Thank you so much. It's really fascinating talk.
Hey.
Hi, hey. Thank you so much. Really, really great talk. I really enjoyed it. So I have a question, because there's a lot of studies actually going back to like losing weight by dietary patterns. And it actually shows that losing weight slower and in a steady rate is more beneficial, because you're not losing so much lean weight and also some other cardiometabolic aspects of this. So it brings me a little bit to the question that maybe just, to as the devil advocates, that maybe the GLP-1 solution is really good because it goes actually in a steady, maybe a bit more slower rate than if we add on top of that something that will go super fast.
Yeah.
And at the end, just asking about the,
It's a great question, and we should deal with that in the fall. But I will tell you is that we don't need to go faster. If you're losing a pound per week, whichever one of these drugs you lose, that's as fast as you need to go. Now, maybe not to the patient, but from a medicinal perspective, that's 50 pounds in a year. Now, can we keep that going so it's 100 pounds after 2 years? That is worth considering. And can we do it with less GI effect? Because quite frankly, whoever worked out these algorithms, 1 month here, 1 month here, 1 month here, they deserve a medal. Because that's not the way we usually develop drugs. That's clinical pharmacologists being incredibly insightful, but it's not user-friendly, all right? We got to get back to some sort of dosing that looks like the way we give drugs, better oral for most, but certainly with an injection where I give you a dose. If you tolerate it and you've got acceptable weight and I want to increase it, then I'll increase the dose. But I don't have to live on this schedule wondering who ends up with a GI effect, has to back up. If I come off the drug, I have to start the titration all over again, right? And so we've gotten into these discussions now. When we lose weight, right, do we lose muscle mass? Are we causing sarcopenia? Are we causing osteoporosis, right? Obesity is a very good treatment for treating sarcopenia and osteoporosis. I don't recommend it, right? But are we going to be developing drugs for fibrosis, for osteoporosis, for sarcopenia, for diabetes, for memory, 6 or 7 different drugs? Or is there an intelligent way to work from above where you can find common mechanisms to treat all of the abnormalities, or more of them, so that elderly subject isn't still taking 7 or 8 medications? So there's a lot there, but again, we've got this is a wonderful problem to manage, right? So thank you for the question.
Thank you once again, and we're happy, Richard, you'll be back in the fall for the sequel.
We'll see.
And we'll learn about the future of this exciting field. Thank you once again, Richard, and thank you for coming.